Chapter 10 is titled Molecular Biology of the Gene. So now we're going to get into DNA replication. DNA replication, remember, that happens during the S phase of the cell cycle, specifically during interphase. DNA replication can be called a semi-conservative process because the two daughter DNA molecules made at the end, they are composed of one original strand, or one, we can say it's a parental strand because it came from the parent molecule of DNA, and then one new strand. So both of them, I see one original strand, one new strand. Because if we look at the overview of the process, what happens is that my original DNA molecule, it unzips. So the nitrogen base pairs are pulled apart, and it's only hydrogen bonds that are linking those together. So now I have two template strands. Well, these template strands will now be used as a guide to fill in the other half. So the nucleus is going to have free nucleotides in it. A is going to pair with T, C is going to pair with G. And that's going to happen until at the end I have two identical molecules of DNA. The blue versus the gray, that is just trying to represent the original nucleotides versus the new nucleotides. They're not actually different colors. Structurally, they are the same. We're going to take a closer look at DNA replication and actually look at some enzymes that are involved. The first enzyme is helicase. From that previous slide, you saw that the first step in DNA replication is that you're going to unwind or unzip, separate the two strands of DNA. That is what helicase does. It's going to do this at places along the DNA called origins of replication. So what you're going to see as the DNA is opening up, you'll see these bubbles, or they're called replication bubbles. As helicase unwinds it more, these bubbles are going to grow in either direction. DNA polymerase, that is the enzyme that will actually read one strand of DNA and add complementary nucleotides. It can only do this to the 3' end of the growing DNA strand. So what I can see in the gray, this is my new DNA strand that's being built. So really all DNA polymerase is doing is it's matching A's with T's and C's with G's. Because DNA polymerase can only add nucleotides to the 3' end, the DNA grows in a 5' to 3' direction. So what's going to happen is as these replication bubbles are opening, because I'm having DNA replication occur in both directions, is that when these bubbles finally merge, that's when I'm going to have DNA replication complete, and I will have two identical daughter molecules of DNA. Another function of DNA polymerase is that it proofreads DNA during this process, and what it's going to look for is incorrect base pairing. So if DNA polymerase accidentally matches A with G, it will actually cut out the G and replace it with a T. This process happens very quickly, and even though there is a built-in proofreading mechanism, mistakes still do happen. Something strange happens as a result of this 5' to 3' direction, and again, it's because DNA polymerase can only add new nucleotides onto the 3' end. You get two different strands being made differently, so we can call one strand leading, one lagging. So let's see what's going on. Okay, here I'm looking at a replication fork, which is really just one part of the replication bubble. So as my parental DNA is opening up, and the parental DNA, it's being shown in blue, this one is called the leading strand, because if this is the 3' end of my parental strand, then that means this is going to be a 5' end of my new strand being built. So here DNA polymerase can attach, and then it can just build from 5' to 3', and it can build continuously as this replication fork opens more and more. But I have a problem with the lagging strand. The lagging strand, I can't build this way as my DNA is opening up. I can only build 5' to 3'. So what's going to have to happen is that as the replication fork opens, DNA polymerase will attach, and then it'll synthesize to the end. Then it's going to have to let go. It'll backtrack. It'll attach, synthesize to the other fragment, and then again let go. And then it'll bind here, synthesize to the end, and then let go so that it's still synthesizing in this 5' to 3' direction. So now on this lagging strand, I get these short pieces of DNA that are called Okazaki fragments. I need to get those pieces linked together. So DNA ligase, that is the enzyme that is going to link together these short pieces of DNA on the lagging strand.